know that living beings grow, and to grow, they need various things. Sunlight, water, and soil nutrients are essential for plants to grow. Human beings too need food and water to grow. We can also say that growth is one of the fundamental and basic characteristics of all living organisms. Growth is an irreversible increase in size of an organ or its part or a cell, which in turn leads to the growth of the individual or organism. We see a seed turn into a seedling, which further grows into a plant and finally into a fully grown tree. We must know that there are several factors which control growth of plants. Certain chemicals released by the plant itself also affect its growth pattern. These are known as plant growth regulators. In our class today, we will discuss about plant growth regulators or plant growth hormones.
Hello everybody. I welcome you all to our today's class. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਅੱਜ ਦੇ ਟੌਪਿਕ ਦਾ ਅੰਦਾਜ਼ਾ ਹੋ ਗਿਆ ਹੈ। ਬਿਲਕੁਲ ਠੀਕ। ਅੱਜ ਅਸੀਂ ਪਲਾਂਟ ਗਰੋਥ ਰੈਗੂਲੇਟਰਸ ਜਿਵੇਂ ਸਾਈਟੋਕਾਈਨਿਨਸ, ਇਥਾਈਲਿਨ ਅਤੇ ਐਬਸਿਸਿਕ ਐਸਿਡ ਐਟਸੈਟਰਾ ਬਾਰੇ ਡਿਸਕਸ ਕਰਾਂਗੇ। ਇਹ ਇੰਡੋਲ ਕੰਪਾਊਂਡਸ ਵੀ ਹੋ ਸਕਦੇ ਹਨ ਜਿਵੇਂ ਕਿ ਇੰਡੋਲ 3 ਐਸੀਟਿਕ ਐਸਿਡ, ਪਾਵ IAA, ਡੈਰੀਵੇਟਿਵਸ ਆਫ ਕੈਰੋਟੀਨੋਇਡਸ ਜਿਵੇਂ ਕਿ ਐਬਸਿਸਿਕ ਐਸਿਡ, ਪਾਵ ABA, ਟਰਪੀਨਸ ਜਿਵੇਂ ਕਿ ਜਿਬਰੈਲਿਕ ਐਸਿਡ, ਪਾਵ GA3 ਜਾਂ ਗੈਸਸ ਜਿਵੇਂ ਇਥਾਈਲਿਨ ਦੇ ਰੂਪ ਵਿੱਚ ਹੋ ਸਕਦੇ ਹਨ। ਪਲਾਂਟ ਗਰੋਥ ਰੈਗੂਲੇਟਰਸ ਨੂੰ ਫਾਈਟੋਹੋਰਮੋਨਸ ਵੀ ਕਹਿੰਦੇ ਹਨ। The PGRs can be broadly divided into two groups based on their functions in a living plant body. One group of PGRs are involved in growth-promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. ਇਨ੍ਹਾਂ plant growth regulators ਨੂੰ plant growth promoters ਵਜੋਂ ਵੀ ਜਾਣਿਆ ਜਾਂਦਾ ਹੈ ਜਿਵੇਂ ਕਿ auxins, gibberellins ਅਤੇ cytokinins। ਕੀ ਤੁਹਾਨੂੰ ਪਤਾ ਹੈ ਕਿ plant growth regulators plant ਵਿੱਚ ਬਹੁਤ ਹੀ ਮਹੱਤਵਪੂਰਨ ਰੋਲ ਅਦਾ ਕਰਦੇ ਹਨ। The second group of plant growth regulators are plant growth inhibitors, ਜਿਹੜੇ ਕਿ inhibiting activities ਲਈ ਜ਼ਿੰਮੇਵਾਰ ਹੁੰਦਾ ਹੈ ਜਿਵੇਂ ਕਿ dormancy ਅਤੇ abscission। Plant growth regulator abscisic acid belongs to this group. The gaseous plant growth regulator ethylene could fit in any of these two groups, but it is largely an inhibitor of growth activities. In general, it is accepted that there are 5 major classes of plant hormones based on their structural similarities and their effects on plant physiology. Each class has positive as well as negative functions. These 5 major classes of plant growth regulators are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. We have already discussed auxins and gibberellins in the previous classes. So today we will discuss about cytokinins, ethylene, and abscisic acid. Let us start with cytokinins. Cytokinins have specific effects on cytokinesis and were discovered as kinetin, which is a modified form of adenine, that is, one of the purines we studied in the structure of DNA. Carnitine does not occur naturally in plants. चलो साइटोकाइनेंस दी होंद बारे विजुअल्स राही कुछ समझिए.
The first naturally occurring cytokinin-like substance was identified in young maize or Zea maize grains. It was therefore named zeatin. A similar form was also extracted from coconut milk, that is also known as liquid endosperm of coconut. While studying their functions, it was found that cytokinins were responsible for rapid cell division, especially in growing tissues. It promotes cell division in presence of auxins. Cytokinins promote cell division even in non-meristematic tissues. In tissue culture, mitotic divisions are accelerated when both auxin and cytokinins are supplied. Cytokinins are responsible for delaying
senescence
or aging of tissues by controlling protein synthesis and transport of nutrients. Natural cytokinins are synthesized in regions where rapid cell division occurs. For example,
root apexes,
developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplasts in leaves, lateral shoot growth, and
adventitious
shoot formation. Hence, it is called anti-aging hormones. Cytokinins also induce flowering in certain species of plants, such as lemna. Cytokinins also increase the resistance power of the plant for temperature variations and diseases. It is also helpful for delay of leaf
senescence.
That's why it is widely used in tissue cultures.
In simple words, ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ cytokinins developing root apexes, developing shoot buds, fruit development ਅਤੇ shoot formation ਲਈ ਬੜੇ helpful ਹੁੰਦੇ ਹਨ. ਜਦੋਂ ਇਹ chemically auxins ਨਾਲ bonded ਹੁੰਦੇ ਹਨ ਤਾਂ ਇਹ highly effective ਹੁੰਦੇ ਹਨ. The proportions of cytokinins produced affect the growth period of the plant throughout its life. Cytokinins, when bonded with ethylene, promote abscission of leaves, flower paths and fruits. But correlation of auxins and cytokinins in the plants is always constant. So, ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ cytokinins cell division, shoot formation ਅਤੇ development of leaves ਵਿੱਚ help ਕਰਦੇ ਹਨ. ਆਓ ਹੁਣ ethylene ਬਾਰੇ ਜਾਣੀਏ. Ethylene is a plant growth regulator. It is produced by almost all plant organs, but its maximum production occurs in ripening fruits and in senescence of leaves. Ethylene functions as an inhibitor as well as a promoter, but largely it is a growth inhibitor. Let us explore more about ethylene.
Ethylene regulates many physiological processes as it increases the girth of the plant and promotes horizontal growth. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of fruits. That is why it is used to stimulate ripening of bananas, apples, mangoes, citrus fruits, tomatoes, etc. These fruits are picked and packed before ripening, and subsequently ripening is regulated with ethylene as and when required. Actually, the ripening of fruits by ethylene is due to the increase in the rate of respiration. This respiratory effect is called
climacteric
effect. Ethylene is also responsible for flowering and budding in pineapples. It also induces flowering in mangoes. It promotes apogeotropism in roots, means it acts as an agent for negative geotropism. That is why seedlings develop epicotyle hook. Ethylene increases
senescence
of leaves and flowers. It includes abscission of leaves, flowers, and fruits. It breaks seed and bud dormancy, initiates germination in seeds
and in
stored foods like potato tubers. Low concentration of ethylene induces root elongation, growth of lateral roots, and root hairs.
So we can say that ethylene plant which respiration ਦੇ rate ਨੂੰ promote करता है. Ethylene breaks seed and bud dormancy, initiates germination in peanut seeds and sprouting of potato tubers, etc. Ethylene promotes rapid internode and petiole elongation in deep water rice plants, that it helps leaves and upper parts of the shoot to remain above water. Do you know that newly germinated seedlings produce more ethylene as compared to a mature plant? Let us know more about it.
When a new shoot is exposed to light, it produces a signal for ethylene production in the plant. Ethylene affects cell growth and cell shape. It prevents cell elongation, causing the stem to swell. The resulting thicker stem can exert more pressure against the object, slow down its path to the surface. So the shoot does not reach the surface, and the ethylene stimulus becomes extended. Finally, it affects the stem's natural geotropic response, which is responsible for upright growth that allows the plant to grow.
Do you know that most widely used compound as source of ethylene is natural ethaphone? Ethaphone in an aqueous solution readily is absorbed and transported within the plant and releases ethylene slowly. Ethaphone speeds up fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. It helps in thinning of cotton, cherry, walnut. The process of thinning means the removal of some parts of a plant to make room for healthy growth of other parts. Thinning is required to optimize fruit size and quality in orchards. Thinning also helps keep trees from biennial production and maintains a balance between vegetative growth and fruiting. Apples can be thinned chemically by hand or by a combination of both. The primary advantage of hand thinning is that poor quality or damaged fruit can be removed. It also promotes female flowers in cucumbers, thereby increasing the yield. To sum up, we can say that ethylene is a simple plant growth regulator. It is synthesized in large amounts by tissues undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedlings. Ethylene is highly effective in fruit ripening. Ethylene regulates hormones such as abscisic acid or stress hormones. Now let us discuss about abscisic acid.
Abscisic acid is also called ABA. This plant growth regulator is composed of a chemical compound which is normally produced in the leaves of plants, especially when plants are under stress. In general, it acts as an inhibitory chemical compound that inhibits the plant metabolism and thus affects the bud growth and seed germination. It is found in freshly abscissed leaves. It plays a key role in natural falling leaves and seed dormancy. Therefore, in several plants, when abscisic acid level decreases, the growth occurs due to increase of gibberellins. In absence of abscisic acid, most of the buds and seeds start growing. Abscisic acid again accumulates within seeds during fruit maturation, preventing seed germination within the fruit or seed germination before the proper season. In aquatic plants, abscisic acid plays a key role in closing the stomata. Soon after plants are water stressed and the roots are deficient in water, a signal moves up to the leaves, causing the formation of abscisic acid. From leaves, abscisic acid moves towards the roots, which is translocated to the plants through the vascular system. It modulates the potassium and sodium uptake within the guard cells, which then loses turgidity, resulting in closing of stomata.
ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ ਐਬਸਿਸਿਕ ਐਸਿਡ ਭਾਵ ABA ਡੋਰਮੈਂਸੀ ਅਤੇ ਐਬਸਿਸ਼ਨ ਦੇ ਰੈਗੂਲੇਸ਼ਨ ਵਿੱਚ ਮਹੱਤਵਪੂਰਨ ਰੋਲ ਅਦਾ ਕਰਦਾ ਹੈ। ਪਰ ਦੂਜੇ ਪਲਾਂਟ ਗਰੋਥ ਰੈਗੂਲੇਟਰਸ ਵਾਂਗ it also has other wide-ranging effects on plant growth and development. ਇਹ plant growth ਅਤੇ plant metabolism ਦੇ general inhibitor ਵਜੋਂ ਕੰਮ ਕਰਦਾ ਹੈ। Abscisic acid seed germination ਨੂੰ ਰੋਕਦਾ ਹੈ। ਇਹ ਵਿੰਟਰਸ ਵਿੱਚ bud dormancy ਨੂੰ ਪ੍ਰਮੋਟ ਕਰਦਾ ਹੈ। ਇਹ ਸਟ੍ਰਾਬੇਰੀ ਵਰਗੇ ਕੁਝ ਸ਼ਾਰਟ ਡੇ ਪਲਾਂਟਸ ਵਿੱਚ ਫਲਾਵਰਿੰਗ ਨੂੰ ਇੰਡਿਊਸ ਕਰਦੀ ਹੈ। ਇਹ ਸੀਡ ਡਿਵੈਲਪਮੈਂਟ, ਮੈਚੂਰੇਸ਼ਨ ਅਤੇ ਡੋਰਮੈਂਸੀ ਵਿੱਚ ਇੰਪੋਰਟੈਂਟ ਰੋਲ ਪਲੇ ਕਰਦਾ ਹੈ। ਇਹ ਕੈਰੋਟੀਨੋਇਡਸ ਦੀ ਸਿੰਥੇਸਿਸ ਕਰਕੇ ਹਰੇ ਸੰਤਰਿਆਂ ਨੂੰ ਪੀਲਾ ਕਰ ਦਿੰਦਾ ਹੈ। ਐਬਸਿਸਿਕ ਐਸਿਡ ਐਪੀਡਰਮਿਸ ਉੱਪਰਲੇ ਸਟੋਮੇਟਾ ਦੇ ਕਲੋਜ਼ਰ ਨੂੰ ਸਟਿਮੂਲੇਟ ਕਰਦਾ ਹੈ ਅਤੇ ਹਰ ਤਰ੍ਹਾਂ ਦੇ ਸਟ੍ਰੈਸ ਨੂੰ ਸਹਿਣ ਲਈ ਪਲਾਂਟ ਦੀ ਟੋਲਰੈਂਸ ਸ਼ਕਤੀ ਇਨਕਰੀਜ਼ ਕਰਦਾ ਹੈ। ਇਸ ਲਈ ਐਬਸਿਸਿਕ ਐਸਿਡ ਨੂੰ ਸਟ੍ਰੈਸ ਹਾਰਮੋਨ ਵੀ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। Abscisic acid helps seeds to withstand desiccation and other factors unfavorable for growth. In most situations, abscisic acid acts opposite to gibberellins. ਅੱਜ ਅਸੀਂ ਜਿਨ੍ਹਾਂ plant growth regulators ਬਾਰੇ ਗੱਲ ਕੀਤੀ ਉਹ ਹਨ ਸਾਈਟੋਕਾਈਨਿਨਸ, ਇਥਾਈਲੀਨ ਅਤੇ ਐਬਸਿਸਿਕ ਐਸਿਡ। ਚਲੋ ਹੁਣ ਪਲਾਂਟ ਗਰੋਥ ਨਾਲ ਰਿਲੇਟਡ ਕੁਝ ਹੋਰ ਟਰਮਸ ਨੂੰ ਸਮਝਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰੀਏ, ਜਿਵੇਂ ਕਿ ਡਿਫਰੈਂਸੀਏਸ਼ਨ, ਡੀ-ਡਿਫਰੈਂਸੀਏਸ਼ਨ ਐਂਡ ਰੀਡਿਫਰੈਂਸੀਏਸ਼ਨ। ਸਭ ਤੋਂ ਪਹਿਲਾਂ ਅਸੀਂ ਡਿਫਰੈਂਸੀਏਸ਼ਨ ਬਾਰੇ ਗੱਲ ਕਰਾਂਗੇ। ਜਿਵੇਂ ਕਿ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ ਨਵੇਂ ਪਲਾਂਟ ਸੈਲਸ ਰੂਟ ਐਪੀਕਲ ਅਤੇ ਸ਼ੂਟ ਐਪੀਕਲ ਮੈਰਿਸਟੈਮਸ ਤੋਂ ਬਣਦੇ ਹਨ। ਇਹ ਬਣਨ ਤੋਂ ਬਾਅਦ ਵੱਖਰੀ ਵੱਖਰੀ ਸ਼ੇਪ ਲੈ ਲੈਂਦੇ ਹਨ ਅਤੇ ਅਲੱਗ-ਅਲੱਗ ਕਾਰਜ ਕਰਦੇ ਹਨ। ਭਾਵ ਉਹਨਾਂ ਦੀ ਡਿਫਰੈਂਸੀਏਸ਼ਨ ਹੋ ਜਾਂਦੀ ਹੈ। ਡਿਫਰੈਂਸੀਏਸ਼ਨ ਨੂੰ ਸਮਝਣ ਲਈ ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ differentiation is a process in which cells derived from apical meristems and cambium differentiate and mature to perform specific functions. Differentiation ਦੀ ਮਦਦ ਨਾਲ ਇੱਕ developing stage ਵਾਲੇ embryo ਵਿੱਚ embryo cells ਜੋ ਕਿ unspecialized cells ਹੁੰਦੇ ਹਨ multiply ਹੋ ਕੇ specialized cells ਵਿੱਚ ਬਦਲ ਜਾਂਦੇ ਹਨ। ਡਿਫਰੈਂਸੀਏਸ਼ਨ ਦੇ ਪ੍ਰੋਸੈਸ ਦੇ ਕਾਰਨ ਹੀ ਬਾਡੀ ਦੇ ਡਿਫਰੈਂਟ ਆਰਗਨਸ ਬਣਦੇ ਹਨ। ਚਲੋ ਇਸ ਨੂੰ ਐਨੀਮੇਸ਼ਨ ਰਾਹੀਂ ਸਮਝਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰੀਏ।
Differentiation. In plants, new cells are formed at two main regions where meristematic cells lie. When they divide, they keep on moving away from the meristematic regions. Once they leave the meristematic regions, they become differentiated according to their position in the plant organ and their function at maturity. During differentiation, cells undergo many types of change, both in their cell walls and protoplasm. For example, for the formation of
tertiary elements,
the cells lose their protoplasm and develop strong elastic lignocellulosic secondary cell walls, enabling them to carry water to long distances.
ਇੱਕ ਹੋਰ ਟਰਮ ਹੈ ਡੀ-ਡਿਫਰੈਂਸੀਏਸ਼ਨ। ਇਹ ਇੰਟਰਸਟਿੰਗ ਫਿਨੋਮੀਨਨ ਪਲਾਂਟਸ ਵਿੱਚ ਵਿਖਾਈ ਦਿੰਦਾ ਹੈ। ਕੁਝ ਖਾਸ ਕੰਡੀਸ਼ਨਸ ਵਿੱਚ ਲਿਵਿੰਗ ਡਿਫਰੈਂਸ਼ੀਏਟਿਡ ਸੈਲਸ ਜੋ ਆਪਣੀ ਡਿਵਾਈਡ ਹੋਣ ਦੀ ਕੈਪੈਸਿਟੀ ਗੁਆ ਚੁੱਕੇ ਹੁੰਦੇ ਹਨ ਡਿਵੀਜ਼ਨ ਦੀ ਕੈਪੈਸਿਟੀ ਨੂੰ ਰੀਗੇਨ ਕਰ ਲੈਂਦੇ ਹਨ। ਇਸ ਫਿਨੋਮੀਨਨ ਨੂੰ ਡੀ-ਡਿਫਰੈਂਸੀਏਸ਼ਨ ਦੇ ਨਾਂ ਨਾਲ ਜਾਣਿਆ ਜਾਂਦਾ ਹੈ। ਇਸ ਦੇ ਵਿੱਚ ਮੈਚੁਰ ਸੈਲਸ ਜੋ ਕਿ ਪਹਿਲਾਂ ਡਿਵਾਈਡ ਹੋਣ ਦੀ ਕੈਪੈਸਿਟੀ ਗੁਆ ਚੁੱਕੇ ਹੁੰਦੇ ਹਨ ਫਿਰ ਤੋਂ ਡਿਵਾਈਡ ਕਰਨ ਲੱਗਦੇ ਹਨ। ਫॉर एग्जांपल, interfascicular and cork cambium are both formed from fully differentiated parenchyma cells. These cells regain the ability to divide to produce more cells. Once these de-differentiated cells are formed, they lose the capacity to divide, but mature to perform specific functions. That is, they get re-differentiated. Overall, we can say de-differentiation is a process where living differentiated cells that have lost the capacity to divide regain the capacity to divide, while redifferentiation is a process by which de-differentiated cells once again lose the capacity to divide but mature to perform specific functions. It must be remembered here that both de-differentiation and redifferentiation occur under certain conditions. एक और term है Vernalisation. चलो उसनु समझ लइए। Vernalisation. Actually, vernalisation is a phenomenon in which flowering plants are either quantitatively or qualitatively dependent on exposure to low temperature. ਇਸ ਫਿਨੋਮੀਨਨ ਨੂੰ ਵਰਨਲਾਈਜੇਸ਼ਨ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। ਭਾਵ ਵਰਨਲਾਈਜੇਸ਼ਨ ਪਲਾਂਟਸ ਦੀ ਲੋ ਟੈਂਪਰੇਚਰ ਟ੍ਰੀਟਮੈਂਟ ਹੈ।
It prevents speedy reproductive development in the growing season and provides sufficient time to plant to reach maturity. Vernalisation promotes flowering in plants at very low temperature. Some important food plants such as wheat, barley, rye have two kinds of varieties, winter and spring varieties. The spring varieties are normally planted in the spring and come to flower and produce grain before the end of the growing season. Winter varieties are planted in autumn. They germinate during winter with small seedlings and continue growth in the spring and are harvested usually around midsummer. Another example of vernalisation is seen in biennial plants. A biennial plant is a flowering plant that takes 2 years to complete its biological life cycle. In the first year, the plant grows leaves, stems, and roots or vegetative structures. Then it enters a period of dormancy over the colder months. Usually the stem remains very short and the leaves are low to the ground, forming a rosette. Many biennials require a cold treatment or vernalisation before they will flower. During the next spring or summer, the stem of the biennial plant elongates greatly. The plant then flowers, producing fruits and seeds before it finally dies, such as sugar beet, cabbages, carrots, etc. These are some of the common biennials.
ਸੋ ਅਸੀਂ ਇੱਥੇ ਜਾਣਿਆ ਕਿ ਕੁਝ ਪਲਾਂਟਸ ਨੂੰ ਲੋ ਟੈਂਪਰੇਚਰ ਦੀ ਲੋੜ ਹੁੰਦੀ ਹੈ ਜਿਸ ਨਾਲ ਅੱਗੇ ਚੱਲ ਕੇ ਉਹਨਾਂ ਦੀ ਫਲਾਵਰਿੰਗ ਇੰਪਰੂਵ ਹੋ ਸਕੇ। ਇਸ ਟ੍ਰੀਟਮੈਂਟ ਨੂੰ ਵਰਨਲਾਈਜੇਸ਼ਨ ਦੇ ਨਾਂ ਨਾਲ ਜਾਣਿਆ ਜਾਂਦਾ ਹੈ। ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਵਰਨਲਾਈਜੇਸ਼ਨ ਬਾਰੇ ਸਮਝ ਆ ਗਿਆ ਹੋਵੇਗਾ। ਇਹ ਤਾਂ ਸੀ ਸਾਡਾ ਅੱਜ ਦਾ ਟੌਪਿਕ। ਚਲੋ ਹੁਣ ਅੱਜ ਅਸੀਂ ਜੋ ਕੁਝ ਸਿੱਖਿਆ ਹੈ ਉਸ ਨੂੰ ਰਿਵਾਈਜ਼ ਕਰ ਲਈਏ। There are 5 major classes of plant growth regulators, which are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Cytokinins root apex, shoot buds, fruit ਦੇ development and shoot formation ਵਿੱਚ ਬਹੁਤ helpful ਹੁੰਦੇ ਹਨ। Cytokinins ਨੂੰ anti-aging hormones ਕਿਹਾ ਜਾਂਦਾ ਹੈ। इथाइलिन एक गैसियस प्लांट ग्रोथ रेगुलेटर है। इथाइलिन फ्रूट राइपनिंग ਲਈ ਬਹੁਤ ਇਫੈਕਟਿਵ ਸਿੱਧ ਹੁੰਦਾ ਹੈ। ਇਥਾਈਲਿਨ ਫਰੂਟ ਰਾਈਪਨਿੰਗ ਲਈ ਰੈਸਪੀਰੇਸ਼ਨ ਰੇਟ ਨੂੰ ਇਨਹੈਂਸ ਕਰਦਾ ਹੈ। We also learnt about vernalization, dedifferentiation and redifferentiation. ਹੁਣ ਸਮਾਂ ਆ ਗਿਆ ਹੈ ਅੱਜ ਦੇ ਟੌਪਿਕ ਨੂੰ ਖਤਮ ਕਰਨ ਦਾ। क्यों ना एक छोटे ਜਿਹੇ ਟੈਸਟ ਰਾਹੀਂ ਤੁਹਾਡੀ ਨੌਲੇਜ ਚੈੱਕ ਕੀਤੀ ਜਾਵੇ। ਪਹਿਲਾ ਕੁਐਸਚਨ ਹੈ which plant growth regulators are known as anti-aging hormones? The answer is cytokinins are known as anti-aging hormones. My next question is which plant growth regulator is known as a stress hormone? Answer: Abscisic acid is known as a stress hormone. Next question. Which plant growth regulator enhances the respiration rate during ripening of the fruits? The answer is ethylene enhances the respiration rate during ripening of the fruits. The next question is name the process in which cells or tissues undergo a change towards a more specialized form of function, especially during embryonic development. The answer is differentiation. My next question is name the phenomenon in which flowering plants are either quantitatively or qualitatively dependent on exposure to low temperature. The answer is vernalization. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਅੱਜ ਦਾ ਵਿਸ਼ਾ ਜਾਣਕਾਰੀ ਭਰਪੂਰ ਅਤੇ ਮਨੋਰੰਜਕ ਲੱਗਿਆ ਹੋਵੇਗਾ। ਅੱਜ ਲਈ ਇੰਨਾ ਹੀ, ਫਿਰ ਮਿਲਾਂਗੇ।